Thanks. Well, it is really an honor to be here. Kind of shockingly, this is my first visit ever to the MVZ, and given the kind of roots I have here through academic lineage, you know, it's kind of surprising. My advisor, Tom Smith, his advisor was Frank Patelka, who did one of the great bird monographs on Phylogenomics jays. Carl and I talked about how we're going to be pronouncing Phylogenomics, Aphelocoma differently. I guess Frank pronounced it Aphelocoma. I've heard this. So that monograph has been nightstand reading for me for the better part of 10 years. And then Alden Miller, there were times during my dissertation where I literally felt like, or I literally was walking in his footsteps. Some of his work in the Sierra del Carmen, one of these remote mountain ranges in northern Coahuila, was one of my major study sites for my dissertation. And some of the observations he made there about niche expansion in birds kind of formed the initial thoughts and basis for one of the chapters of my dissertation. So lots of good roots at the MVZ, and very excited to be here, look over those field notes of Alden Miller's. Part of my visit here also, I'm interfacing with Carla about getting the Moore Labs collection up on VertNet. And many of you might not know, the Moore Lab has a large collection of birds. They have over 60,000 specimens actually, and the vast majority of that is Mexican material. So we have a lot of birds. They've never been online and searchable in a database, so we're working on that. It's a lot of skins, and also I'm pretty sure at Occidental College, with only 2,000 students, we're right there up at the top in terms of the ratio between skins and students. We might be number one in that. So today I'm not talking too much about collections, but what I do want to emphasize before I get started is that collections, natural history museums, have been very important to this project. All of the specimens you'll see are vouchered museum specimens and natural history collections. And really this project kind of came out of museum research, museum scientists, and people in organismal biology. So I think that's important to emphasize in these times of budget cuts and some museums being under stress. Here at the MVZ, you've never had any problem convincing people you do big science, because you do, but hopefully we can kind of export that to some other of the smaller natural history collections. And I hope this is a step in that direction. So it is an exciting time to be working in a museum. It's an exciting time to be an evolutionary biologist, because we are in this kind of crazy transition, revolution in terms of our DNA sequencing technologies. Going from the capillary sequencer at 3730 in terms of the number of reads you can get, moving down to a 454 and a Illumina HiSeq. Obviously we're moving exponentially, and the price per megabase is dropping exponentially. I don't know if any of you saw this latest gizmo that was unveiled. The Oxford Nanopore plugs into your USB, sequences DNA in real time. They say it produces 40 kilobase reads. You know, if it even lives up to a part of this hype, you know, this is another revolution on our hands. I'm pretty sure it also syncs with your iTunes. So it's just getting, I mean, it's almost bewildering. It's been fun to watch the rapid pace, but at the same time, I think for our field, and here I'm talking mostly about phylogeography, phylogenetics, and population genetics, it's been a bit of an uneasy time as we sort of try to figure out exactly how we can harness that technology. We know the technology is out there, and we've just been trying to figure out how we can use it. Our fields have some specific needs, so we need orthologous DNA sections, so sections with a shared ancestry. That's kind of what it's all about. We're comparing those orthologous sections between individuals, populations, and species. We prefer to have lots of different independent pieces of DNA to address the fact that, you know, we know now that genes can have different histories, and it's not really usually those different histories we're so interested in. It's the species history, and so the species history is sort of an amalgam of all these different gene histories. So looking at a lot of them is important. And we prefer to use a whole lot of individuals. This is kind of something that's not specific, but you know, it's to our field and a few other fields. You know, sometimes we want to look at over 1,000 individuals, some of those really, really intensive phylogeographies or population genetics studies. And you know, you don't see this in every field. So we've got these three things that we want. And right now, these goals are being met more or less by sequencing PCR amplified loci using conserved primer pairs. This is kind of how we've been doing it and how we're still, in most cases, doing it now. The problem comes in, you know, we've got this increasing machine capacity. And it's going up, certainly not linearly, it's exponential. Yet the human capacity is still sort of stuck in this PCR phase, in this locus discovery phase, trying to find out what conserved primers you might be able to use. So as you continue working with PCR and churning up loci, you're sort of getting the feeling like maybe you're not using all the capacity that you could. So the idea is, how can we get this human capacity to sort of match with our machine capacity? So specifically, how, you know, what we want is we want protocols that are scalable to the machine output. So we want something where if we add more individuals, we add more loci. It's not an incremental work that we're doing. We want something that's not so difficult to just add in more loci and add in more individuals. So we want something like this. And then we want the specific markers that we're using to be collected from many genomic regions, and we want them to be as universal as possible. And by that I mean they're going to work across different individuals, different populations, different species, and maybe even very deep taxonomic scales. And that just kind of reduces that time on the locus discovery end of things. You want things that are just going to be broadly applicable. So before I go further, I want to introduce the collaborative team, my collaborators on this. It's very, very much a collaborative project. And I can really say that without any one of these individuals that I'll introduce you to, you know, probably this project would never have gotten off the ground and gotten to the stage it's at now. We've got three parts to it. One is sort of the wet lab protocols for the high throughput sequencing, kind of prepping the samples to be ready to go on one of these fancy new sequencers. And here, Travis Glenn at University of Georgia, he's kind of our wet lab guru. On the bioinformatics side, and we have this kind of data, you need a lot of bioinformatics. We've got Brant Faircloth, he's a research scientist at UCLA, just a complete data wizard. And Nick Crawford, a graduate student at Boston University, who's also been chipping in on the bioinformatics. Never met Nick in person, which might tell you something about kind of the collaborations that we're beginning to form in this kind of new world we're in. Hopefully I'll meet him at the Evolution meeting this year. We've traded probably 1,000 emails. And then, and I really purposefully put us down here at the bottom because I almost feel like, you know, we're perfunctory in some ways, is the experts with the phylogenetics and the species trees. And this is where I come in, and Rob Brumfield is kind of figuring out how we take this data once we get it, and we start, you know, getting the finished product of answering the questions with the questions. So the methodological breakthrough that's kind of forms the foundation for what we're doing is a method called sequence capture. Many of you are probably familiar with it. If you're not, I'm just going to walk you through the basics. Essentially, it's a method that seeks to be essentially a PCR replacement for these new sequencing technologies. And how it goes is basically like this. You have a genomic target, some DNA that you're interested in sequencing. You can design some probes that are complementary to that target. They can be made out of DNA, they can be made out of RNA, the probes. And you put a little piece of biotin on the end of it. This is a vitamin. It's basically just like a structure. You have this on your probe, and it binds to your genomic target. So you can put these probes with the biotin attachment into a bunch of shredded up DNA of an organism, and they'll all kind of go in there and bind to their targets. And then biotin has a very high affinity for a protein called streptavidin. These are kind of like, oftentimes they're associated with beads, so the streptavidin beads that you put in, and the biotin with the genomic target attached to the probe gets attracted to the bead, and allowing you to wash away everything else that you don't want. So all of this other DNA goes away, and you're kind of left with your targets, which then can be released from the biotin. And you can do this with not just one genomic region, but you can do it with multiple. In fact, you can do it with thousands, tens of thousands of targets, all mixed in together, all of the different probes for those targets in a solution, in one solution that you put your DNA in. So you can simultaneously collect thousands of targets from around the genome. And then those targets, the idea is they'll all be sent to the sequencer, the next generation sequencer, to be sequenced simultaneously. That's kind of how the machine, the human output meets the machine output. Well, as we know, nature tends to come in these organism-sized packets of DNA. You can see how far I've come from being a field biologist.
Well, they are Australian.
Are they? But yes. And so you can think of these as like small little pools of DNA, which was great for capillary sequencing, because this is divided up, 96 wells, each individual can go in a different well, but the whole setup of a next generation sequencer is a bit different. Essentially, it's like one big pool that you put all of these things into. So the trick here, and I don't go into really any detail at all, but when I'm talking about the method we go through, this step is always inherent, is that finding ways to tag individuals before you put them on the sequencer. So the way you do this essentially is by affixing little DNA-based tags in front of all those shredded up pieces of DNA that you've targeted with the sequence capture. So each individual gets its own unique identifying sequence so that they can be pooled together, sequenced in the big bucket, and then afterwards you kind of use bioinformatics to sort them and assign them back to the individuals you were looking at. So I won't talk really any more about this step, but it's implicit. Okay, so well, that's two of our wants satisfied. We've got collected from many genomic regions, that's a sequence capture. We can do many, many individuals, as many as you can design tags for, the sequence tagging. And well, what about getting those markers to be as universal as possible, as broadly applicable as we can possibly get them? This classic MVZ paper from 1989, looking at conserved primers in mitochondrial DNA, kind of opened the door to phylogenetics, deep-level phylogenetics at a broad scale. Been cited over 4,000 times. It was pretty epic contribution. And these conserved primers lie on the outside, and in the PCR world, what you were looking at was the variation on the inside. But what we needed, kind of hearkening back to your SAT analogies here, as PCR is to these conserved primers, we need what is it to sequence capture? Sequence capture is to what? The setup's a little different, because we want this probe that's kind of right in the middle. We don't want two little pieces on the outside. So a paper came out in 2003 describing ultra-conserved elements in the human genome. The text is a little wonky here. I thought I might just read a few pieces of it. It's really pretty interesting. 481 segments longer than 200 base pairs that are absolutely conserved, 100% identity, between orthologous regions of the human, rat, and mouse genomes. Nearly all of these segments are also conserved in the chicken and dog genomes with an average of 95 and 99% identity. So at the time they wrote this, they didn't really know what these ultra-conserved elements did. They just knew that they were pretty cool, and they described them. And since then, there's been a whole flurry of research into what these ultra-conserved elements do. And our thinking was, well, we don't care what they do. We wanted to know, what can we use them for? So we took a very utilitarian point of view. And of course, that's not exactly true. We're all very interested in what they do. And what they do probably also matters a lot to how you might use them. But essentially, we just wanted to use them for our probes. So the original discovery was in a three-way genome alignment between human, mouse, and rat. They found these like over 200 base pair conserved regions that were separated by a bunch of unalignable or not very conserved stuff. Brandt's first kind of contribution and brainchild to this effort was doing the same kind of an alignment between the two bird genomes that were available, so the chicken and the zebrafinch. He lined up these two organisms and found that they had a lot of conserved elements too. They also had ultra-conserved elements. He focused on things that were at least 60 base pairs or longer, because this was about the size of the probe that would be used in this sequence capture technique. And then he took these conserved regions he found between chicken and zebrafinch and he blasted them to the anolis genome. So he actually then brought anolis into the equation, sort of expanding these ultra-conserved elements out to amniotes, getting broader. And if he found regions that were less than 120 base pairs, he would take them and buffer them with some flanking sequence from the chicken and design a probe from that. If he found regions that were greater than 180 base pairs, he would design tiled probes to kind of go over that whole, cover that whole ultra-conserved region. So these are the probes that eventually were manufactured from this process. So these are our universal markers. So just to orient you here again, what we're looking at, what we're mainly interested in is what kind of flanking sequence are we going to get. So we have a probe that's going to fall into a very conserved region. You're also going to capture some flanking sequence with all of your DNA, and we thought that here's where the variability is going to be that we're going to use to build our phylogenies. And in fact, kind of giving you a little data in advance here, we did find this to be the case. So this is some data from 9 different bird species from throughout the bird tree of life. And you can see the core UCE sits about right here. This is distance from the core, and the frequency of variant bases moving out from that core element starts to increase almost exponentially. Eventually you get out to the edges and you start getting, you know, all these crazy insertions and deletions and things fall apart. But for a while, just outside that core, things are looking nice and variable and still alignable. In terms of how many UCEs you can find in amniotes, so this is kind of like moving out phylogenetically from birds. If you start with about 5,000 of these UCEs, and this is kind of how many Brandt found in the beginning, you know, you see a drop-off as you move outside of lizards into mammals, so a group that the UCEs weren't even first discovered in by Brandt. You get some drop-off, but not a whole lot. By the time you get out to frogs, so outside of amniotes, you do get quite a considerable drop-off, but there's still over 1,000 UCEs that are shared between frogs all the way over to birds. So that's a lot of potential universal markers in invertebrates and tetrapods. Comparing this to exons, so there's a lot of interest in doing these kind of techniques with exons too. I think exons are great and very useful. However, there's a much steeper drop-off when you look at that kind of comparison. comparison in exons. So this is moving from humans out into more distantly related mammals, and then eventually over here in the birds and anolis, you get a really steep drop-off in the number of exons, whereas in UCEs it's much more gradual. I don't want to try to pull the wool over your eyes here. We've got very different y-axes, a lot more exons out there than UCEs, but in terms of the shape of that drop-off, much more steep in exons. You end up at the end with more UCEs than you do with exons. So okay, there we've got the markers, we've got kind of our three wants satisfied. The process is taking DNA from a critter, shredding it up, adding it to all of these probes. The probe set we tended to use was one that was 2,200 probes. These probes bind to the DNA in various places. We wash away the non-target DNA, kind of just leaving those targets. They come in a bunch of little reads. If you do this on an Illumina sequencer, you get fairly short reads that you then got to align, piece together and align into loci, into contigs, and then you align them into loci. Multiple different individuals aligning each locus. And then from there you can concatenate them and run them in a Bayesian analysis with Mr. Bayes for your phylogeny. Or another thing we've done is run these through a gene tree species tree program. So you can consider each one of these loci separately, build gene trees from them, and then from those oftentimes discordant gene trees, build one species tree. And this is especially important for really rapid divergences. I'm not going to get into it much in this talk, but in rapid divergences, you just get a lot of random gene sorting processes going on, and the genes can have conflicting answers with one another. And you know, the more rapid that divergence has been, the more you're going to see this sort of random gene effects. So you've got to take that into account somehow, and these kind of methods do that. So I'm going to run you through 4 places where we've applied these ultra-conserved elements to phylogenetics. It's all, at this point, deep-level phylogenetics. So one of these is in placental mammal relationships. So all placental mammals fall into one of 3 kind of very large superorders. You've got Boreotheria, which includes humans and rodents and horses and cows and things like that. Aphrotheria, which includes elephants and tenrecs, and then also Xenarthro, which includes things like sloth and armadillo. The speciation scenario here, like which of these superorders is more closely related to which, what was the order of speciation, has been really controversial. You wouldn't believe the number of papers that are out there that are only devoted to figuring out what is the speciation order among these 3 things. And right, there's only 3 possibilities, so it's kind of wild. I mean, there's seriously like 15 papers on this. Most phylogenomic studies point to Aphrotheria and Xenarthra being sister taxa. And this has a lot of biogeographic implications because they're both superorders that are found mostly in the south, so that they've kind of linked this back to how Pangea split up into 2 pieces, with these 2 superorders being on one piece. So it has some biogeographic implications. What we know is that the speciation events there were very rapid. And you can know this for sure by looking at rare genomic changes. Oftentimes for deep events where your DNA sequence data might have a lot of saturation, or you start to get some multiple hits obscuring the old signal you've got in the DNA, it's nice to go to these rare genomic changes because they give you a really clear idea that's not marred by saturation and homoplasy. Even when you look at those, you get conflicting signal for which genomic changes support which of those splits. So taking this into coalescent theory, this knowing how much discord is in those rare genomic changes, it basically tells you that this speciation event is in a part of phylogenetic space that is showing profound gene discord. So just to kind of illustrate this more graphically, what I did was go in and I did some coalescent simulations for genes under some sort of realistic demographic parameters for what that ancient mammal way back when might have been like. So you don't want to make any great predictions there because you don't really know. So I just took a broad swath of things like generation time and what the population size might have been like, and I said, how often are the genes that you would look at going to agree or disagree in their answer about that speciation scenario? And that's plotted on this scale here. So for a speciation interval of 5 million years between those events, you can see that most of the state space down here is in a region of gene discord. And when you actually look at the empirical data of these rare genomic changes and you plot that here, it's way down here. So the empirical data is saying this speciation scenario is in an area of profound gene discord, and it's in fact even really close to an area where if you use concatenation and you treat all of your genes together as if they're just one super gene, it's actually going to give you the wrong answer. So they call this the anomalous gene tree zone. The interesting thing is that nobody thinks this scenario that the speciation interval was even 5 million years. In fact, most people agree that it was probably closer to about 1 million years, and under that scenario, that anomalous gene tree zone just expands, and you know, the upshot is that really you're going to need those species tree methods I talked about to be able to answer this question. It's going to be very difficult, if not, you know, coming to the wrong answer to do it with the concatenation approach. Okay, so we had our ultra-conserved elements that we had identified from amniotes. I've walked you through that. For this study, we designed probes, but they were just like in silico computer-based probes. We didn't actually go in and synthesize the probe itself in DNA or RNA, because there are a lot of mammal genomes that are already out there. So we aligned those probes to the 25 placental mammal genomes that are out there in 4 outgroups, and we cut out a region from the genomes that had 1,000 base pairs of flanking sequence. We aligned them, and if ever it was the case that there was one species that was missing a locus, we just went ahead and dropped that locus completely from the data set. So all the data sets we have are just a complete, full, solid data matrix with no missing loci. At this point, we had absolutely no idea if this was going to work at all, if UCEs and their flanking DNA was even going to produce a phylogeny that was going to make sense. I went in advance of when I was supposed to say that. Once we get those loci, create gene trees for them. From the gene trees, create a species tree, which we did by a couple different methods, and we did a bootstrapper sampling method of the species tree. You're going to see a lot of bootstrap scores up there. I don't think they're comparable to what you might be used to with posterior probabilities and normal maximum likelihood values of bootstrap scores. I think there's some evidence that they probably tend to have lower values, but there's not a lot of work that's been done on that yet. Okay, so then what I said about not knowing what was going to happen. We were delighted to find that the phylogeny we got out of it was, in broad terms, completely compatible with all of the new recent studies, including new data of mammals. So all of the cool, interesting relationships they found in the last 5 or 10 years, for instance, like tree shrew grouping with the primates instead of with the rodents. All of these things were reflected in our species tree. This data just came out in a paper, I think it's just still in advanced online access in genome research. And I want to point out the number of loci we had. So we got up to as many as 917 loci that we could align between these placental mammals. With all of these taxa you see here, we had 183 loci. As you start dropping a few select taxa and going kind of narrower in, you can boost this number of loci. Just to tell you a few things, just a few basic pieces about the data. So looking at variable sites and levels of saturation, just to kind of orient you, you might be more familiar with data from exons. So there's a nice exon data set, Mark Springer and William J. Murphy and their epic mammal work they've done on that phylogeny. They have a nice set of exons that we can compare our UCEs to. And we found that in terms of variability, the flanks of our UCEs were roughly comparable to like first and second positions of codons and exons. Same with saturation. Meanwhile, inside that core of the UCE, that very conserved core, we did find a lot of variation, but it was less, but also quite a bit less saturation in the signal. And then here is third positions of codons and exons. Obviously, those have a high amount of variability, but also a high amount of saturation. So in terms of this question of the divergence order of the superorders, our data indicated that Aphrotheria was kind of the first superorder to branch off from the rest. Meanwhile, Xenarthra and Boreotheria were sister taxa. We saw this in a couple of different data sets. This is kind of data from a stripped down data set of just 7 taxa, 591 loci. The bootstrap support, again, this is species tree bootstrap support, not super high, but more than twice as high as support for any of the other scenarios. So one thing we actually didn't get at very well with this particular data set was the evolutionary origin of bats. We kind of got some conflicting answers with these data, and the question of where bats have their evolutionary affinities has been one of these long-standing enigmas. There was a paper that came out on rare genomic changes about 5 years ago that had a very weird position for bats. They put them in a group with carnivores and perissodactyls, things like horses and rhinos, in a monophyletic group, and then to the exclusion of Sitartiodactyla, so things like cows, hippos, whales, sheep, and pigs. They named this group Pegasopherae, after the winged horse. And it's obviously odd because it splits the hoofed mammals up that are in this particular clade, and obviously has some kind of weird implications in terms of morphology. Well, there's a really recent paper that just came out using 97 PCR amplified loci that actually rejected Pegasopherae. Found kind of evidence for a more traditional placement of bats outside of those hoofed mammal groups. At the same time this came out, we were in the process of mining 12 genomes specifically from this group, so we didn't want to expand it out more because we didn't want to drop a bunch of loci. So we just looked at 12 genomes in the group where bats is. We found 819 UCEs. We actually used exons as well. We found 376 exons. And the phylogeny that we got supported Pegasopherae. So we found bats in their grouping with horse and the carnivores. This is a species tree, still really high bootstrap support. If you just did a concatenated tree with a Bayesian analysis, the support was 100%. So, and then here's the Sitartiodactyl out here on the outside. So, long live Pegasopherae until the next study, potentially, I guess. But with over 1,000 loci, you start to wonder, what else is it going to take to answer this question? So another thing we've been interested in is applying this to the evolutionary origins of turtles. So this is another one where turtles have always kind of been a question in broader amniotes. Where do they lie? Where are their evolutionary affinities? A lot of the early morphological work tended to put them, as a really basally diverging branch of amniotes because of their weird skull morphology. More recent morphological work has tended to kind of nest them within within this group of snakes, lizards, crocodiles, birds, and ally them with snakes and lizards of Tuatara. So diapsids, nest them within diapsids. The genetic work that's been done, there's been a series of studies and kind of increasingly clear from the genetic angle that turtles ally with the archosaurs within diapsids, so with the crocodilians and the birds. However, and this is just this is an example study, probably the best, most thorough in terms of its taxonomic sampling out there that was done and put turtles in with archosaurs. Recent paper came out 2011, again on rare genomic changes, this time not retroposons but microRNAs, that said, hold the phone, no, turtles are actually in a group with snakes and lizards, so with the lepidosaurs. And this kind of had a lot of DNA sequence data, people sort of scratching their heads saying, how could this possibly be? So we sought to answer this question, and this time, instead of just mining genomes with the UCEs, we actually were generating some new data as well. So we went through this whole sequence capture process with the UCEs. We needed a snake, so these are things that don't have genomes. Obviously, we needed a snake. We needed Tuatara. We needed a couple turtles from each of the two very divergent branches of turtles, and 3 crocodilians. We had saltwater croc, American alligator, and a gharial. And then to fill in the rest, we did some genome mining of the anolis genome, chicken, zebrafinch, and human. So we did I think a couple lanes of Illumina HiSeq. Generated 4.9 million reads per species, which were assembled into scads of contigs, but eventually whittling down those contigs into alignable loci with no missing data. We ended up with a data set of 1,145 loci. Average, each of those loci was 447 base pairs per locus. So not too long, but maybe long enough. The answer we got out of that was unequivocal. Turtles ally with archosaurs, with birds and the crocodilians, whether it's a species tree, whether it's a Bayesian analysis of concatenated data, the answer was the same. It was 100% support for that relationship. So we felt like we shored that one up pretty good. Turtles are archosaurs. Well, now we kind of return to something a little closer to home for me, birds. Felt much more comfortable in this element in some sense, but in another sense, this was like the hardest question of them all. Birds had a very rapid radiation, many, many lineages, all at the same time, diversifying a really long time ago. And in phylogenetics, this kind of leads to the perfect storm of phylogenetic problems. You know, you've got a lack of information on those very short internodes early in the phylogeny. There just hasn't been enough time for a lot of mutations to accumulate, so you don't have a lot of data. It's a long time ago, so you've got saturation and homoplasia of your DNA sequence data. And then you've got this problem of the randomness of gene sorting thrown on top of it. You've got a lot of coalescent stochasticity causing gene tree discordance. So overcoming these 3 problems is not easy, and there's been fairly slow progress elucidating relationships among the bird orders and families. And some people have even suggested that maybe the problem is hopeless, it's kind of an unresolvable polytomy. Here we had the help of Mike Harvey, very diligent graduate student at LSU, who not only goes out into the Amazon and discovers new species of birds, but he's also pretty handy with a pipette in the lab. And our idea was to kind of take this fairly recent, fairly awesome new phylogeny of birds by Hackett et al. Also Rory's a co-author on that paper. Told us a lot of new and very interesting things about birds that I won't bore 75% of you with. But we kind of used that as our backbone, and we went in and we sampled 37 bird species that were more or less evenly dispersed throughout that phylogeny. We took 2,200 of our UCE probes, or those ones that we deemed were really good for birds. And we went through our sequence capture protocol, and we multiplexed in 4 lanes of an Illumina. Ended up with over 100 million reads, assembled into 1,200 to 1,500 contigs per species. Average contig length was 395 base pairs, although some of them got up to 600 base pairs. And the average coverage we had on each one of those contigs was 50 to 200 times. So we put together a pretty nice data set. If you looked at all 37 of the species, and again, you just dropped every locus that had any missing data, you ended up with 265 loci. And this was all sequence capture. So for these 265 loci, this is like none of the genome data. If you started to drop a few of the problem taxa, you could boost that number of loci way, way up. So with just 20 of the species, you could have a full data matrix of 816 loci. I want to show you a few just kind of like rough, untrimmed alignments just to give you an idea for what this data looks like, because I think it's kind of interesting. Your average locus looks a bit like this. These are all the species on the side. This is just like a screen capture from Genius. You can see this conserved area where the probe sits down. You do get some variability and even some apparently informative variability in that UCE region, but generally that variation is out there on the flanks until eventually either the data, that fragment is shredded at a certain point and drops off, or it just becomes really like bizarre and unalignable. You've got some alignments that turn out to be very highly conserved, so there's not a lot of variability. You've also got some alignments that clearly show evidence of shared insertions and deletions. Kind of interesting too, we haven't done anything with that data. So the tree, we're getting ready to submit this. It's not quite ready yet, but the tree we've got, this is the concatenated data from a Bayesian analysis. This is the species tree. Again, I'm not going to tell you what's related to what and all that, and put most of you to sleep. If you're interested, I'm happy to go over it taxon by taxon. But what I want to tell you is that we got a very well-resolved neo-aves. So neo-aves is kind of the part of the bird phylogeny that's been the hardest to resolve. And with a few exceptions of nodes here that we've collapsed if they were under a 95 posterior probability, we seem to have a fairly well-resolved tree. Species tree, not so much. Actually, we collapsed everything. I think if it was under 40, we collapsed it. So we actually left some of the things that were above 40 because we thought they might be interesting. But there are some reasons, I think, behind why the species tree did not look very well resolved that I could talk about with you if you're interested.
Can I just ask which method you used for that species tree?
This is STAR. So it's not a very computationally intensive, like Bayesian thing. It's basically like an analytical way to solve a species tree. It'll churn out a species tree like that. It's generating the bootstrap scores that kind of like takes forever. But it'll churn out a species tree just analytically based on coalescence in the gene trees. But what I'll tell you about this also is that it largely validates a lot of the cool novel relationships that were found in this Hackett paper, with a few added novelties. So one I thought was particularly cool. We seem to find consistent support in neo-aves for the pigeons and doves being the most basal divergence of the neo-aves. It seems like pigeons and doves are a really old lineage that diverged from the rest of neo-aves a long time ago. I feel like there was one more thing I wanted to say about that. I can't remember what it is.
Watson.
Oh, Watson. I was actually just going to leave that one. Why did you see that? Yeah, so Watson is this really bizarre looking bird, and there's been all kinds of hypotheses for where its evolutionary affinities lie. There hasn't even been any good molecular study that's come up with a solid answer. we have a really strong support for Watson grouping with Charadriidae, which is a lot of the shorebirds, a lot of water birds, but also includes things like Hasana and other things that maybe you could believe is more like Watson. I'm not going to go to the mat believing that that's absolutely true, so, but it's kind of interesting. Anyhow, okay, well, the conclusions, I hope I've convinced you that ultra-conserved elements are phylogenomic markers that are well suited to sequence capture in high-throughput phylogenomics. The flanking sequence from these UCEs can help resolve some of the great outstanding mysteries in vertebrate deep phylogeny, but I think the question maybe you're all thinking to yourselves is, do they work at shallower timescales? Okay, you've shown us this deep phylogeny. We don't have a lot of data on this right now, but I will show you a graph. This is from the supplementary information of one of our papers. This is showing variability it's showing you the location of Snips in the human SNP map as you move out from the center of these UCEs. So I think what this shows is that even at kind of the most recent level of human population divergence, there are a lot of SNPs that line those UCE flanking regions. From our mammal phylogeny, you know, we went down to like chimp, human, gorilla divergence and got really good results there. So hopefully in between humans and the chimp-human gorilla divergence, we think that UCEs will also be useful for those phylogenetic questions as well. But we're currently working on that. Another question I get a lot is, why not just do whole genomes? And my answer is usually that. I hope to sit around and have a beer with a lot of you while I'm here, and we can talk about that. I think the reality is that when you have whole genome data, alignment and determination of orthology is, I mean, to say it's not trivial is also kind of an understatement. It's very, very difficult. Obviously, much of the genomes of organisms is unalignable at deep phylogenetic scales. And the whole analysis of genome-genome alignments is kind of in its infancy. Meanwhile, we have the tools for analysis of discrete loci. We have this great phylogenetic framework built up. So that is what we're trying to use. Bottom line, genome data work well in concert with the UCE approach. So we've got people that are producing genomes that then want to use the UCEs to mine the genomes. And obviously we wouldn't have the UCEs if we didn't have those original genomes. So we owe a debt of gratitude to those genomic projects as well. So I want to thank Carla and Rory, Craig, and Anna for organizing, for funding this visit. Again, the samples were all vouchered specimens. I thank the collectors and the museums that contributed them. The sequencing facilities that did the Illumina sequencing, and of course the genome folks that put together those initial genomes that allowed us to discover those UCEs. And you for listening. I'd be happy to take questions.
Really great time and fascinating. Can someone hit the lights, please? And then we'll take some questions.
Yeah, of course.
So, with the UCEs, do you know how, two parts of the question. One, how they spread across chromosomes, especially on the, are they on the sex chromosomes?
They are on the sex chromosomes.
And where on the chromosomes are they? Or like in the centromeres or more?
They're all over the place. They're almost evenly spread throughout all chromosomes and even the sex chromosomes. They think that they tend to be in these almost kind of like genomic deserts. But then they also are, some of them overlap with exons. They're kind of just all over the place. Yeah.
So I assume UCEs can get duplicated? I assume UCEs can get duplicated too, so presumably you would also have to define orthology for those UCEs, just as with genomic data?
Right, yes. So you have to determine orthology, but there is some previous work that says that UCEs have very little overlap with parts of the genome that tend to have gene duplications and paralogous genes. So we do a scan for duplicates, and we drop any locus that seems to have like weird duplications that we didn't expect. And then we think that in general UCEs are not really in gene duplicated regions. But there's clearly a lot more work that could be done. But I think the whole determination of orthology is easier in UCEs than it is in things like exons. Would be my guess.
So I have a question about the sequence capture method that you're using.
Mm-hmm.
So with these probes, you're obviously you're going to hybridize your target DNA, but you're also going to get some DNA that I imagine is not related to your probes. And I assume you're trying to strike a balance here, right? Because if you only get DNA that exactly matches your probe, that's not going to be very useful. But when you're dealing with this DNA that's not, this kind of junk DNA or this DNA that just comes through, so how much of that are you getting, and is it easy to identify it, and does that mess with, do you have problems with your analyses, assembly and other things, and kind of identifying it and dealing with it?
Yeah, we get a ton, right now we're getting a ton of DNA that's non-target. So some of that bird data, 80% of the reads actually will be stuff that doesn't align to the probes. And you just throw that out, which is kind of unbelievable. This, like tens of millions of reads. You're just like, oh, see you later. But then once you've done that, so you kind of blast your probes that you expected to get against the reads that you got. You just send the rest of it off, and then you're focusing on the stuff that's the probe. So it's, I can't say that it's trivial, and if Brant was standing here right next to me, he'd be like beating me over the head because I'm making it seem like it's so really easy. I mean, it's something that takes him a lot of time to weed out the trash from the target. But it doesn't present a lot of problems downstream once you get it out of there. Was there another part of the question that was in there? Okay.
Yes.
Just curious to know how you're planning on dealing with library prep costs so you can expand this out to hundreds of individuals as opposed to 30.
Mm-hmm. Yeah, because that's the major expense right now is these birds we did with Nextera kits, which are about, if you get a good deal, you could probably get one for like $1,500 that does 20 samples. However, Travis and Brant have been working on some methods to kind of homebrew that whole library prep process, and I think they're onto some things that are, you can use it with standard NE. You can do it with standard NED reagents. It's going to bring that cost way down. You're not going to need a kit. So I imagine whether it's them or some other group, the protocols are going to emerge really quickly to bring the cost down on that library prep step. It's the most expensive step. The sequencing is not really that expensive given what you get out of it. Bottleneck in terms of cost is the library prep.
So I have one brief question. I mean, you built a fabulous consortium of folks there, and I think quite a few natural history museums and also other just food to do systematics are doing this and perhaps developing some things in parallel. But do you have any thoughts about how we can develop sort of larger consortia that are developing these methods?
Yeah, I don't. I mean, I haven't put a lot of thought into that because we've been so focused on just like getting our stuff out there. But I think what we're finding as we talk to more people is, there's so much overlap, and each group has a little bit that they've kind of perfected that you can add in. All of our stuff is open source. All of our code is posted online. I know Brant and Travis are really big believers in open science and open source, so they're always interested in collaborations. But yeah, it's something that we need to think about so that everyone's not reinventing the wheel every time we try to do
particularly the case with bioinformatics.
Yeah, exactly, because these are all custom scripts.
A lot of nuances to that.
I know, if Brant decided he wanted to move to the moon or something like that, I mean, it's like, I don't know what would happen, so.
I'll take one more, then maybe a couple.
Just a much more technical question. I mean, in some ways, it's very similar to some of the stuff we're doing in the lab now with bait capture, but our problem was always the barcoding of the probes. So I'm curious, how did you barcode that many probes? is it custom design, all those barcodes, to be able to tease so many of them out, or are you using?
We add them after. So we have the probes, we do the library prep, then we do the sequence capture, and then after we do them separate by individual, the sequence capture, I mean the sequence capture itself is nothing, it's just like some stuff in a plate that sits on a PCR machine for 24 hours. And then afterwards, then we barcode them individually, and we've got some custom-made adapter, like PCR-based primers, where you add in the barcode afterwards in a PCR step.
So you're barcoding by locus and individual, or just by individual?
Just by individual.
Yeah, so it makes,
that's,
makes all the difference.
Yeah, exactly. So, right, then afterwards you don't have to do it by locus because they'll just
But that should get much harder as you get closer phylogenetically, don't you think? If you're pooling individuals.
Well, when we pool individuals, they're still, by that time they're barcoded by individual. So, yeah, we don't have.
Yeah, maybe not.
Let's take one more question.
I was wondering, you have show that very nice genius alignment of all the individual species, but you don't actually get, that snip in one line. You get like a frequency spectrum, right, of the different changes in, if there is a G or a C there, you get, 80% G, 50% or 20% C. So how do you bioinformatically like turn that into an alignment that you can then send into?
Right. So
Snips from sequencing errors, right?
yeah, exactly.
Yeah.
So right. So so this, in terms of like calling something a SNP versus a sequencing error, we have enough coverage that when something's like really low frequency, that just gets weeded out. So the sequencing error is taken care of. When you have a genuine heterozygote, how do you call that? And right now, because we're doing such deep phylogeny, we're not worrying about it. So we're just saying any differences between the two copies in an individual is going to be dwarfed by the differences between the individuals, in our case. However, if you start applying this method to more recent stuff where coalescence becomes important, you need to phase those two strands, we need some methods to be able to do that, because we're stitching it together from little pieces, right? So you can't just phase the loci, you just don't know which goes with what. So I'm not sure what the answer to that is. Maybe you could then supplement what you have with some longer 454 reads that are going to allow you to phase, because you get longer fragments all in one.
Are you using ambiguity codes, or are you just randomly calling these?
I think they're just getting randomly called. So we end up with just like one of the bases in a heterozygote, and we're just saying it doesn't matter for this question, but obviously that's not ideal. I don't think it's a problem for our results in this case, but as we get down to smaller and smaller scales, it's going to become a problem.
Okay, well, I'd like to have you join me in thanking John for